and eukaryotic cells. And what I've been really trying to harp or hone in on is what allows prokaryotic cells to grow at an exponential rate. So Jada, can you tell me one thing that we've discussed that would allow a prokaryotic cell to grow more quickly than one of your cells? They don't have a nucleus, right? So not having a nucleus is advantageous. Why, Taylor?
I cannot tell you, I'll be honest.
That's okay. I appreciate your honesty. Who do you think knows?
I think Michaela knows.
No, I was pointing to Shiny Hat.
Oh, okay.
It's like a question magnet, right? Every time I gaze this way, it catches my eye, and I have to ask you a question. What do you think? Yeah. So if you don't have a nucleus, right, and Jada's right, you can divide more quickly. Why?
Because you don't have to break it down.
You don't have to break it down during mitosis. Remember, prokaryotic cells go through binary fission. They lack a nucleus. And so if you don't have a nucleus, you don't have to break it down in order to divide. What else? Give me another one. Jenna? Ooh, yes. Explain that word to me, please.
They can transcribe multiple genes at one time. They don't have to go all the way through the
Yes, right? Good. I just had to have you explain that to me because Ashlyn's article had like 50,000 words in it that I didn't understand, and I'm still trying to process them, right? Good, right? Polycistronic, right? So multiple genes. Remember, any function in your body typically is not going to, well, your body or bacteria is going to require more than one gene to contribute, right? If you're polycistronic and you do all of those genes in one swing, then you're able to do or perform that function much more quickly instead of waiting for individual gene products to be made. Yes, ma'am.
When we're talking about why
Oh, yeah, we are, because it's all part of that process, right? Because, growth is replicating for them, right? What's allowing them to grow and utilize nutrients, right, are typically enzymes or proteins that they're making via transcription translation, right? So yes, so this is a grab bag, and I can almost guarantee you, Alanis, look at me. You see me winking? Holmes, do you see me? All right, see that? Good. That there's going to be a question about this on the exam. And some of you know and probably hate me for the fact that I nitpick at little details, right? But, ooh, wow. That's Taylor. I was expecting you to be like, no, right? Right. I want to see, right, that big picture, all right? And so I really am. The big picture here is that exponential growth, right? And so I want you to be a little comfortable with that, and then you can kind of work your way down for those nitty-gritty details. Ooh, do you have another one for me? I have an answer. Yeah, so there are some eukaryotic cells that have the ability to be polycistronic. The ones that I've looked up, and again, I haven't done a lit search recently to see if anyone has discovered some other ones, but there are some protozoa that have been shown to have it. So yes, there are some eukaryotic cells that do have the ability, right? But the majority don't, and we talk about us, we say we don't, okay? And you'll see in a second, we'll actually talk about mRNA processing here in a little bit, why it doesn't quite work out for us to kind of do it that way. All right, good question. All right, who can give me another thing that we talked about that would allow cells to grow exponentially? I have an answer. Cistronic. But honestly, if you put polycystonic on an exam, I'm not going to get you for it. I'll know what you're talking about, so don't get caught up over that R. Sarah, you want to help me out? Yes. One circular chromosome, right? So you're only making a copy of one, although we've seen it is a very big chromosome. Sarah, what else when it comes to replication of that chromosome allows that to move along more quickly? Yes, bidirectional replication, all right? Remember the math problem that Abby answered correctly. If you have two trains, one at 50, one at 30, moving towards each other, they move towards each other at 80 miles an hour, actually, right? And so moving towards, right, allows you to do something much more quickly. All right, who can give me one more, and then I'll leave you alone, and we'll move on. Now, you've already had your time. No. Oh, you have a question? Okay. Yeah, because your question is going to turn into some awesome answer. I know, I can feel it, right? Caitlin, help me out. What's one more thing? Oh, there should be one more juicy nugget in those notes. Maybe it has to do with transcription. Ooh. Oh, Rachel, we'll let Rachel go, and then Holmes.
I don't have this is what you're looking for, but they have the sigma factor and we have the transcription.
Okay, so it is a difference. It doesn't increase speed, but that's an important. You're close. All right, so my sigma factor binds to my promoter, right? It brings RNA polymerase in to my bacteria, and then why? Ooh, I wasn't even thinking of that one. Yes, no nucleus means you don't have to send your transcript out. All right, they can all happen in the same place, and you'll see that it's going to get a little even more crazy and wacky here in just a second, right? So again, they're not sending their transcript out through those nuclear pores. So transcription, translation, same area. Who can finish off what I was thinking for with Rachel? Read my mind, J Buck. Yes, so as soon as RNA polymerase that bound because of the sigma factor, props for remembering that, right? As soon as it moves off that promoter, another RNA polymerase comes in and can immediately bind to, right? And so you think about it, right? You have multiple transcripts that are being formed at the same time. Awesome. So you guys have already kind of mentioned some of these, right? No nucleus. Holmes just pointed this one out to me, right? Now, some of these are just differences. Some of these are differences that contribute to that exponential growth. I think you guys can kind of discern, right? So here, the start codon for a prokaryotic cell actually makes formal methionine, which is a slight derivative of methionine. That doesn't make it go any faster. It's just the difference. Now, here's one, and I'm sorry that I forgot to mention it. Maybe that's what the 2 slides ago I wanted to do. But this idea of multiple ribosomes. So polyribosome can be translating a strand can be translating a transcript at a time, right? So not only do you have multiple transcripts coming off, right, of a strand of DNA, as soon as those transcripts start to form, you have multiple ribosomes that can then begin binding to those transcripts to start translating, all right? So the rate at which you can make proteins and which you can make enzymes is just staggering compared to what our cells have to do. All right. I'm sorry I didn't bring up a Sharpie. I'll bring it up on Friday to make kind of my drawing here. But what I typically would draw is a strand of DNA going straight out, then your transcripts coming off, and then do you have one? Oh, many. Yes. I'm kind of in a pink to red mood. Oh, right in between. Thank you. All right. Thank you, Jada. So, right, if this was your DNA, right, you have multiple ribosomes that bind, or not multiple ribosomes, I'm sorry, multiple RNA polymerase. And so, right, as they move, you have transcripts, right, that are coming off as they continue to move along, right? Continues building them, they move along. Then, right, you have multiple ribosomes that can bind. So then you have those coming off here, and you can see, right, the rate and speed, right, the efficiency is just incredible, right? You have to make a transcript, you have to send it out of the nucleus, right? It goes to a ribosome in the cytoplasm or a ribosome in the rough ER, and then it does its job, right? It's such a slow process in us compared to them. Now, please don't take away from eukaryotic cells. Thank you so much. It still isn't, well, actually, let me get a, let me be nice just in case. I don't want this to be known as the dry erase board marker incident that started a rampage through the science department. All right. So again, really, you guys have a lot of different things going on, right, that you can kind of relate back to for these prokaryotic cells growing so much more quickly than our cells, right? And again, this is just brief. This is certainly not complete, right? Just saying, hey, we have to send our transcript out of the nucleus where it's translated by ribosomes. Again, this doesn't affect speed, but our starting amino acid is slightly different. And again, one ribosome translating, it does its job. And then another one comes in, right? You don't have multiple ribosomes translating at the same time, right? So you have things for replication, transcription, and translation. And, right, you can kind of relate to all of those things, location of some of these events that are contributing to the overall speed, right? That exponential growth. Now, here is, another important difference. And you guys have probably heard this or learned this before if you've taken previous biology classes that have talked about cells. But when a protein is made, right, that typically is never the final product. There's some type of post-translational modification, right? And so in some cases you have proteins that help fold things and put things in place, all right? Now, in prokaryotic cells and eukaryotic cells, part of that amino acid sequence is helping direct that protein and telling it where to go, right? Again, think about the speed, though. In order for our, proteins to be modified, right, we want to add sugars and lipids, just like prokaryotic cells do. But again, ours have a specific place, right? So you have to have transcription, translation, and then, right, you move your way through the ER and you can have these sugars or these protein, or these sugars or these lipids, added. Again, everything's in the cytoplasm of a prokaryotic cell. And so while you're finishing that translation, right, you can actually start modifying it with the addition of lipids and sugars. So it doesn't have to go someplace special. It can all happen right there. All right, so let's talk about one of the things that makes us a little slow. And this goes back to, I think, Jada's kind of question about polycistronic versus monocistronic. All right. So your mRNA, right, has a methylated guanine, right, on the 5' end cap. It also has a poly A tail, right, where we add approximately 200 adenine derivatives, right? But importantly, right, we have introns and exons, right? So remember, the way I kind of keep these straight is introns are intruders. And what do you want to do to intruders, Michaela? What do you want to do to intruders? Get them out. Good. There we go. I can work with that, right? Yes, splice them out. All right. And so here, right, you have your exon and your intron. You want to remove those introns, and then you actually have, right, your mRNA. So we have pre-mRNA, right? So this is another kind of tidbit or piece of information you can put into your brain about why do prokaryotic cells grow so much more quickly. When a prokaryotic cell, I'll get you in just a second, Taylor, I promise. When a prokaryotic cell, right, makes a transcript, that mRNA is ready to go. It doesn't have to have that methylated guanine. It doesn't have to have the poly A tail put on it. It doesn't have to have any introns removed. It is ready to go, right? Your mRNA is not. You actually make pre-mRNA. We modify it, right? Chop it up, glue it back together, and then we can translate. All right, so we have mRNA modification that takes place before translation. They don't, right? So again, they are saving time. You also, right, can kind of see from this would make our cells capable of polysystromy. I don't know what the verb or the adjective there is, but you know what I'm trying to say, right? It would be very hard for our cells to be capable of being polycistronic because of this mRNA processing, right? It just doesn't work as well for us. All right, Taylor, lay it on me.
Okay, so if an intron is an intruder, what exactly is an exon? Is it just like a normal part of the eukaryotic?
Yeah, so I don't really get into this too much just because I hate to call it, the term used to be when I was in high school, and even early on in college, is this idea of junk DNA, right? So that intron, we'd call it junk DNA because we really didn't know what it contributed to. There's now research showing that that might not be the most accurate way to describe it. There seems to be some purpose to that. I haven't checked into the junk DNA dilemma, but the exon is then going to be the functional gene unit, all right? And so once you have this mRNA here, I would say this is a functional gene, right? And so this is part of it, this is part of it, this is part of it. But if you were to just translate, I'm sorry, if you were just to transcribe that and then get this RNA, because it's broken up, you wouldn't get a functional gene if you immediately translated that. So it's kind of, you're putting the puzzle together. It'd almost be like, ooh, you seem like someone that likes murder mysteries, right? If I gave you like, 3 pieces of the map, right? I cut it up into 3 pieces and I gave you one. Well, that's useless, right? You need all 3 of those pieces put together to read it. And that's kind of what's going on with our genes. Does that make sense? Okay, perfect. All right. So, again, this is everything that I just said. Some of you prefer tables over me. I got it. I'm not offended. So if you need to look at a table to kind of help tie some of this mRNA stuff together, it's right here. But this is talking about differences in transcription translation. Again, some of these differences are simply cosmetic, right? Like that formal methionine, that's not increasing the speed at all. It's just kind of a biological fact, right? But we do have things, right, that affect the speed at which prokaryotic cells can grow. Okay. We've already kind of mentioned this earlier on a slide where I showed you, it just had this diagram had said, hey, gene A is only turned on when we need it. And then I think it had like gene C that be constitutively expressed, right? It's always on. And so again, this right here is not any different between prokaryotic cells and your cells, but it's still something interesting to talk about, right? Is, why are we expressing genes, right? And I'm not trying to go into some deep philosophical discussion or debate about life, right? But ultimately what it can come down to, if you really want to break it down for me, is I would say that we express genes for metabolism, right? We need energy, right? We need to harvest energy and then we need to build things, all right? Now, when it comes to harvesting energy, some genes are more important than others. And so, steady state metabolism. This is when I talked about last lecture, and I know it confused some of you with this idea of a housekeeping gene. If you're doing an experiment, you need to have a baseline of what gene expression is. And so you choose a gene that you know is always on. Someone in here, Michael, was it you that talked about actin? Is that you? Remember? Oh, that was Hannah True, one of my other Western blotters, right? So Hannah used actin in a Western blot, right? That's a protein that's always expressed because it's an important component of your cytoskeleton. All right, so you could even do that, right, on a gene or DNA level. You could use actin as your housekeeping gene because it's always being produced. This is GAPDH. Those of you that have had biochem maybe have nightmares about GAPDH, but this is an important enzyme in glycolysis, right? Glycolysis is really important. You want to be able to harvest energy, so you always have this gene on. This is the same in prokaryotic cells and eukaryotic cells. So we have housekeeping genes that we could use in a bacteria that we could isolate, and then we can do something to that bacteria and compare the expression of those genes. Now, we're going to see some of the same effects in us, but it is much easier to read, right, or detect changes in prokaryotic cells when they experience stress, danger, right, abundance, famine, right, all these different things in their external environment, right? You, right, have a very complicated, we'll say, physiological system that handles all of these things, right? So if I'm looking at the effects of these things in you, I am studying multiple systems to figure this out. Prokaryotic cells, right, it's a single cell, right? And so we are able to easily measure the effects, right, of these things. So we can see what genes are turned on during stress, danger, abundance, famine, right? We talk about abundance and famine. You guys have some basic knowledge of this, right? If something's developing into an endospore, right? Remember we said Well, hey, it doesn't just form endospores for fun. It forms endospores because it notices that the environment is not favorable, right? You know, abundance, you guys actually did really, really well. I was super happy. I think I've graded through that question on your last exam about biofilms, right? We'll actually talk about this here in just a second. Biofilms, remember, you had all those different bacteria coming together in one environment. Well, that improves their ability to communicate, all right? And the reason they can communicate is because they start releasing different proteins that allow them to talk to one another. But they're only going to do that if there's a lot of them around, right? It's not advantageous to be sending out signals if you don't have people to receive the signals, right? Ooh, look at that. Prophetic when it comes to my slides, huh? Or maybe I've just done them too many times. So this is the biofilm that we talked about, right? Can someone remind me? I mean, just for kicks, why not? On the biofilm question, I was looking for 3 things. I actually got a couple other answers that I thought were good, and so still got most of the credit for. But I talked about biofilms being important for 3 kind of main things. Savannah, can you give me one of the things? Well, what does it look like they're doing with one another? Communicating. Okay, good. Communication, right? Good communication. What's another thing that biofilms are important for? Protection. What's another? Nutrient sharing, right? And some of you put on the exam, and I actually like this one too. I kind of grouped this with protection a little bit, right? But allowing a bacteria to occupy or inhabit an environment that it couldn't if there wasn't a biofilm, right? We use the example, and you guys, I think, did a good job of hearing this, that in your lab settings, right? That's like it's the perfect place for bacteria, right? That Petri dish with your agar in it, or your tube with your broth in it, right? That's not the real world, and so forming a biofilm, right, allows them to attach, adhere to, and live in certain environments, right? And so signal transduction, right, and we'll talk about this a little bit more. We might just end here, and then we'll pick up again on Friday, but is basically this idea that bacteria start to produce, right, certain proteins that allow communication, right, this signaling event to occur. The sad thing is, right, again, if you're by yourself and you're producing these signals, they don't find anybody. There's nowhere to go. But in a biofilm, when you start dividing, right, because you found a good environment, you start attracting other bacteria, right, because remember, they can live with one another because they sometimes utilize different nutrient sources, right? So it allows this diverse community to form. They start talking, and then these signals, right, they find other bacteria, all right? Some of you that are like really nerdy and maybe are, you know, into sci-fi, and you've seen like those big satellite dishes that they're, you know, sending signals out, hoping to communicate, you know, with aliens. They're not hitting anything, right? And again, we're not going into conspiracy theories. That's one of, I think, 2 reasons. Either, right, there are no aliens out there, or they've been watching 2020 and they're like, just don't want any of this business, right? So either way, right? But you need, right, quorum sensing, right, is dependent here, cell population and density. This critical mass, you need enough members of a community present for those communication signals to find a target, right? And biofilms provide the perfect place for this type of signaling, all right? A dense population with protection, that is an excellent, excellent environment for communicating, all right? We're gonna end here. We're gonna talk very briefly. This slide is not meant to be overwhelming. I think when we walk through it, you'll see that's not that bad, but it's just going over the basics. What happens, right, when a bacteria receives a signal, all right? So with that being said, please remember, we're gonna do an exam. I'll double check either that Friday or that Monday, not November 4th, all right? There'll be a 100-point exam, and then your final will just be 100 points, right? So we're gonna separate those out, hopefully maybe make that a little less stressful. That's my intention. If that doesn't work out, well, Melissa, right? She's my TA, she's fine. Okay, I'll be working on those exams. Thank you very much for your patience and your attention, and I will